the feedback is pretty loud from all the sound coming in. So if you can keep it down, that'd be great.
Yeah, and remember for your remote members, we've already put on Slack live streaming. So we're now live streaming. It's just screen capturing here and capturing audio from here. So when you're presenting, please make sure you're standing here. So your voice is also captured for the viewers over YouTube. Okay. So we'll start, I guess, by talking about just quickly about the sponsors. Sorry, yes.
Question about it. So they won't be able to see the presentation.
No,
okay.
No, yeah.
They see the slides.
They see the slides.
Yeah.
Yeah. Yeah, I think that's the best we could figure out at the moment. So yeah. Do you want to? Oh yeah.
And first of all, this event would not be possible without our great sponsors. They made the Chipotle and the beer yesterday possible, just for you guys. Especially EcoScope for the venue, as well as we have a representative from Accelera, Manny in the back. You want to wave? If you want to talk to him, feel free. And yeah, we'll get this started with a bit of prizes first.
Hello, everyone. Sorry for, I guess, rallying you guys to finish on time today, but let's keep it going. So I saw some like scolding looks, but it's okay. Nothing personal, right? I would just like to start by saying thank you all for coming and thank you for being part of HACKSEQ18. It's, seeing you guys present like work and like it was like really intense, it looked like. Maybe we just give you all a round of applause for doing a really great job. Right. There, we'll start with giving away a few of the t-shirts. A few of the team leaders wanted to single out some kind of exceptional people that they saw throughout HACKSEQ18. So I think maybe we can get Vina up. Vina? Do you want to? Yeah. Oh, that's okay. So do you want to maybe say a quick word about? Oh, do you want to say a quick word maybe about who you nominated?
Yes, I can't remember all the words I want to say, so give me a second. So I nominated Tristan Slater for the t-shirt. It's a very coveted t-shirt award, right? So I really appreciated the way that he works with the team and how he was always positive and upbeat, even when there were issues with our software. We changed a couple times to change the programming language, and he just like kept up. He was always upbeat. He was always excited about like what we were doing, how we were going to go forward, and he was always helping out other team members when they were having issues. Never lost his temper. Really great guy. So I was really happy to have him on my team, and I really look forward to seeing what he's doing for the rest of the year.
Thank you, Tristan. Do you want to come up and grab a t-shirt? And then I think Alex had a few people.
Yes. You want to come here?
Yeah,
sure. All right, I don't have my exact words prepared, but I'd like to nominate 2 of my team members to receive a t-shirt. The first of which is David Mayhew. He flew in all the way from Philadelphia to join our hackathon this weekend and did an amazing job working on our visualization step, our outputs, and making sure that our data looked really clean. And my second nomination is for Benjamin, who also flew out from the East Coast of the United States. He did a great job with our compression stage of our pipeline and was an absolute beast with software engineering and did a great job organizing our logo for our project. So round of applause for these 2.
And I think Rob had someone nominated as well. Yeah, do you want to?
So I nominated GU, and I think G, is very patient with me. I was trying to walk him through some of the hard stuff that we're doing. We were very heavy on the Python programming, and I gave him minimal instructions. We worked really well with Michelle, and then produced something really cool. And he also was really good at making us laugh, especially towards the end today. We all really enjoyed his humor, so I think he deserves a t-shirt.
Do you want to have the people?
Yeah, do you guys want to, while we do the by haiku? I think that we might be out. If there are any t-shirts left at the end, then yeah, we can discuss that in the Slack.
So please come pick up your t-shirts
yeah.
over here, people who got nominated from right here.
You probably want to come down and get your size while the size is available, or you'll end up with an extra large pajamas. Okay. So next, we had the Bio Haiku contest. Thank you everyone who entered the Bio Haiku contest. We had 38 entries, and the competition was really, really stiff. In the end, I think, had 6 of the organizers and Manny from Epsilera do the judging, and we just did rank order. And there was just a few people really vying for that top position. It was incredibly close. We had to swing back and forth between the top 2 a couple of times. So without further ado, oh, I don't know. So we have, is this, what's up? Okay, I see. If you just go to the top, oh, it disappeared. So, sorry, I guess that. Okay, so first we'll start with the Twitter kind of award, or the Twitter favorite. So Sean Jackman, I think you get a t-shirt because Twitter really liked your haiku. So round of applause to Sean's. And then now I forgot who it was. Sorry. Okay, and then the runner-up prize was for Tamar A. Where's Tamar here? Yeah, come on up. Oh, there it is. Do you want to read out your haiku?
Yeah, I guess.
Do I have to?
Come on.
Okay.
We're all friendly. You want to stand here? I think this is you.
This is so embarrassing. For pause in sequence, if pause matches reference, return pause not snip.
I guess, Sean, you want to read out yours?
Yeah, absolutely.
What
What did I say? I said, well, tiny pieces of DNA flow through a pore, we assemble them.
And there you go. Okay, now the grand prize winner. So this is going to be for a DNA ancestry kit. So maybe we get a drum roll going. And the winner of the Bio Haiku contest is David Guao. Come on up, David. Okay. Do you want to read out before?
Yeah, of course. Took me a very long time to come up with this one. So it goes, not ravioli, not spaghetti code. I said pasta, not pasta.
We can talk later about getting everything organized.
Thank you so much.
All right. Thank you all for who submitted a haiku. It was great. We're thinking next year we may do limericks. So polish off your poetry hats for next year, and we'll think of another really creative prize. Thanks again. And next up is Jen.
Yeah. So,
do you want to stand here?
So just while Bara's setting up the first presentation, I just want to let you guys know how the presentations are going to work. So we're going to go starting from 1 to 8, right?
Yes.
Yeah, so we're going 1 to 8, and you each get 4 minutes max. Sasha will be sitting right there in the front, and she will hold up a sign, tell you when your first 3 minutes are up. So she'll warn you at that point, and then at 4 minutes, she will play a very loud noise, and we will cut you off right then and there, wherever you're at. Sounds good? All right, so I guess first off, let's start with group number 1.
Hey guys, I'm Sam from Reuse, and we're presenting rapid elimination of useless sequences. So first off, huge credit to my amazing team. I can't even describe the work they did. So why did we make reuse? First of all, most of you in your sequencing processing need to do some kind of pre-processing, and that involves some kind of filtration of data, whether that's removal of host sequences, removal of contamination, removal of adapters, ribosomal RNA, you name it. K-mers are fast, that doesn't need to be said. Here are the existing solutions. Ours is at the top. You can see that it's downright amazing. You know, compared to aligners, it's much faster. And then the other ones, they're not multi-threaded. You can't store the index to disk, so they're lacking some very key features. Here's what we made. It's just a brief overview on how we did it. So we had 3 threads running, an input, a filter, and an output worker. And then within the filter worker, the big component is that BB hash filter, which is a perfect minimal hash. It's low on memory, so we can hopefully fit it into 8 GB of RAM. It's fast, and then we can filter your sequences. We started evaluating it today. We got some pretty cool stuff. First of all, the leading programs, so things like BBDuck, Cookie Cutter, we were maxing out RAM on 50 GB machines. BBDuck actually maxed out on 115 GB, so really not practical to filter via K-mers. This is how much we're expecting reuse. We were pretty close to getting a final memory assessment, but didn't quite get there today. And finally, you can see BWA is the typical aligner if you want your DNA sequences, and it takes much, much longer than everything else. So in conclusion, you can download it. Just go to our GitHub page, open it up, and go for it. It's ready for you.
Whoever's speaking would want to be closer to the laptop, so 4 people remote to hear.
Okay. Okay, go. All right, we're team 2, Team Awesome Assemblies. So assemblers are usually these monolithic pipelines of modular components, but they're never really, those components are never swapped between assembly pipelines. So that's what we tried to do today, to make chimeric assembly pipelines to make hopefully a better assembler. All right, so we did a lot of genome assembling today, and these are some of the pretty genomes that we assembled. And these are, we started with just off-the-shelf assembly pipelines, so this is kind of our baseline of standard assemblers. And some of these are short read assemblies, some are long read assemblies. And then we started making Franken assemblies. So we did make our chimeric assemblers, which we're pretty excited about.
Yeah, so the way that we made components for these various assemblers is that some of us took on snake-make, some of us took on some really weird and hacky methods called flowcast and assembler flow. And ultimately, we got these really cool and weird assemblies that you have here. So SPABIS is spades and abyss, bcalm times abyss, or bcalmbis, as I like to call it, is that one. And then we have, I don't really know how to make a portmanteau out of that one, but yeah. Should I pass it on? You guys want to talk? No? Okay, you can take it back.
The long-read assemblies were done by our French participants. We had participants in 4 continents, which I think is pretty darn exciting too. And this is another short-read, long-read hybrid assembly using Abyss and links. This was by our participant in India, Swapna. And finally, we did make our leaderboard of awesomeness, which was like comparing all the assemblies together. So an assembly, you want it to be like highly contiguous and have few misassemblies. So here, misassemblies is on the x-axis and contiguities on the y-axis. So you want the best assemblies are in the top left corner. One assembly I want to highlight is in the top right corner, which is one of our Franken assemblers. It has the best contiguity. So it's got some misassemblies, which if we can iron those out, it's got the best contiguity, which is pretty exciting. So we've got a little work to do, but if we move it to the top left corner, that would be the best assembly, which is pretty cool result. Here, say something. Okay, leaderboard. You made the leaderboard.
All right,
Come over here.
I made the leaderboard with the our Shiny application, basically it's just the links at the bottom. It's just a website that currently displays, I guess, this table that we have that tells you about the comparative properties of all these different assemblies. Yeah.
Okay, do you guys want to say a quick thing about what you worked on?
I was making a Cambridge Uni modular component for Flowcraft. There's just a couple of bugs to fix up, but almost there.
I made a user-sector component for the snake workflow system, and as we see, it's dead last right there.
Cool. And the last thing I'll say is that we had 1 pull request accepted to Bioconda, and we had 4 pull requests accepted to Flowcraft, which is pretty exciting that I think this work had some impact outside of our own little repository. So yeah, so participants all over the globe, lots of fun assembling. It was a great time.
Oh yeah, and that's us.
That is our wonderful foreign artist.
Well, I have a presentation in the form. We also have the journey, which you have it. It's in the fog. Here. Is it on there? It's here. Okay, cool, thank you. Okay, yeah, so we are Team Viral Voyager, and you'll see why soon. Okay, so the background, so there's an estimated, I don't even know, what number is this again? Nonillion virus particles in the ocean. And ocean viromes drive biogeochemical cycling through metabolic and gene transfer networks, mainly between bacterial host and the virus. So for that reason, they're major contributors to host diversity, population dynamics, and they produce a very large amount of organic material in the ocean. So just, yeah, a little intro. There are several sequencing projects going on, namely the Terra project, that's the biggest one right now, aiming to understand these metabolic pathways and see how they're being influenced by viruses, and in turn, how the oceans are affected by them. But the data from the projects is heavily scattered and they're inconsistently analyzed. So the Hallam Lab, which is affiliated with Ecoscope, provided us this data set, which they've pretty much unified as many of these several sequencing projects and kind of uniformly analyzed them. So what they did was they assigned metabolic pathways to these open reading frames, which were identified through metagenomic sequencing. And then the quantity of the open reading frames determined the pathway abundance level. So what were our goals? Really, really quickly, we only have 4 minutes. Create a comprehensive repository of ocean virome data sets. That's something that's ongoing. Make this data accessible to both the public and researchers. Create a website, which is what we did for HACKSEQ. And then also perform some type of initial data analysis to be used for publication. Okay, so I'm going to quickly show you guys the website. Yeah, okay, this is it. This is our welcome page. This is the most important part though of our website is our team. These guys, other than me there in the corner, pretty much did everything to produce this website. So, oh crap! Sorry, Kevin. I'm sorry. There he is. He's there. Kevin, yeah. So if you guys ever need a Shiny app, ask any one of these people. You might get a slightly better one though with Ogun. Just joke. Okay, yeah, so this is our main feature. It's a map, which is right here. It's there, it's coming. What was that? It's like halfway.
Too much refresh.
Yeah. Oh, right. Yeah, okay. So we got this cool, beautiful map here, which you can see all the samples that we have collected in our database. So if you, for example, click here, you'll see all the samples that were collected in this region, and you can get some sample metadata about them, like the salinity, the temperature, when it was collected, and most importantly, what pathways were identified there. And the user can, of course, filter the database however they please and then download that data. A really cool part is we've included an analysis tab, so you can kind of compare groups of samples. So for example, I will compare the viral samples, create a group that ends up in the corner there, and then I can compare them to bacterial samples because we do have a bit of bacterial samples to allow that kind of comparison to happen. But you don't have to see. Okay. Yeah, so you can select them, the two groups that you want to analyze. You press analyze. The first thing you do is generate a heat map, which is coming. So yeah, super cool heat map. You can compare the pathways that are coming up in the virus samples and the bacterial ones. So you can see in the virus ones, we've got a lot of pathways coming up. I'm not done. No. And then I'll leave you with that.
Thank you so much. So team 4, yeah, Barry. So, yeah.
Are you ready? Yep. All right. Thank you, Bart. So we are Team Barry, and we're comprised of me, myself, Rob Gilmore, Bruno Grande, Hamid Younesi, Kristen Bystrom, Michelle Lee, Santina Lynn, Sharita Hutchins, and Ziju. So for our project, we use ZenHub to manage everything. It was really helpful. This is mostly just to explain to you the hierarchy of our project. We're called Barry, but we have 3 separate command line tools that we have not yet connected to a centralized command line tool that we're calling Barry. We do have RM, rinse, and rut up today. What those are, REM is a virtual environment manager, sort of like Python's virtual environment manager. So if you've ever used pipenv or virtualenv, that sort of does the same thing, but for R. Rinse is a R installation command line utility. It helps you install R from source on the command line. And then you have rut, which is a utility package for R, which helps you install R packages like sort of like pip or npm. And then you also have some other sort of utilities there that help you do stuff with R. Since we don't have a lot of time, I'm just going to kind of go through and just give you a demo real quick. So let me log into our DigitalOcean server. 1 second. All right, so I'm going to just show you what the CLI looks like. Will you hold this for me? Thank you. So you have RM, which is virtual environment manager. Here's the CLI and the parameters. You have rut. Here's the CLI and the parameters. You have rinse. Here are those as well. So if you want to install something, you can say rinse, or if you want to install R from source, you say rinse install. And oh, sorry, rut install. Am I doing this right, guys? All right, well, I know rut, for instance, will install packages for sure. So it's rut install cran. See, doesn't work. Cran rlang. So now it's installing stuff into the global directory. I'm going to go ahead and try RM. So RM-D. We want our thing to be called HACKSEQ for life. So then it creates an environment variable, and then you do source, HACKSEQ for life, then /activate, and then it activates your environment, and then you can go to /HACKSEQ for life,
/bin/r, and now you're in the environment, and we created a really cool R profile script for R, and it helps you like install stuff quickly. So this is going to install bioconductor in my my virtual environment. So there's your virtual environment for R. And you can actually, hang on, you can actually use BIOC installer to install whatever you want. Come on. BIOC, come on. So EdgeR. And it's going to install EdgeR too. So we just installed BIOC Lite, and we also installed EdgeR with BIOC Lite afterwards.
Awesome.
And that's it. That's it.
Yeah, perfect.
Thank you very much.
Oh, thanks to you. I guess team 5, which you're now Snap, right? Yes, we have changed names. Okay. And thank you very much. All right, so we are project number 5, originally called Alignment-Free Pathogen Genomics, and we have developed a tool called SNAC, which stands for Sequence Non-Alignment Compression and Comparison. So why do we want an alignment-free tool in the first place? Well, our goal is to determine similarity between bacterial sequences. This is important for epidemiological purposes. One example would be tracing the source strain of some sort of disease outbreak. For pathogenic organisms, techniques that use sequence alignment, such as BLAST or muscle, tend to not work very well. This is due because pathogenic organisms sometimes lack sequence homology, or we don't have any a priori information about this. So there's need for some other tools out there, and these tools are alignment-free. There's been some existing alignment-free tools out there, such as MASH, and we have developed one that is called SNAC. So what separates SNAC from other alignment-free tools out there is that we use data compression in our pipeline. So this is a high-level overview of how our pipeline works. We take in sequences as our input, run your favorite data compression algorithm on it, turn out some math, which is listed down below, and we output a distance matrix. Using this distance matrix, we can compare our results using clustering algorithms and obtain some sort of biological context. Whatever this biological context is up for the biologist to decide, but we have a streamlined pipeline that works very well. It works in parallel, so runs really quickly on a computing cluster, and outputs really good set of distance matrix. So some of our results. This is an example of one of our log files rendered in HTML. So we have runtime for analysis, our compression methods used. We have a bunch of optional arguments, such as taking the reverse complement of a DNA sequence. And we get a distance matrix. Our example here uses 2 mystery genomes from Mycobacterium tuberculosis, as well as our dependencies that we use. And next up, we benchmarked it using existing alignment-free tools. The one I talked about earlier, MASH, is plotted on the x-axis, and our tool is plotted on the y-axis. And our p-value showed that our results have a significant correlation to MASH. So essentially what this means is that our tool works really well and compares equally to existing tools that we have, which is really nice. So we had a really big group for our project, and I want to give them a really big round of applause. Thank you. Thank you for sticking to less than the time. So I guess project 6, right?
So the tool we worked on was for simulating structural variants in our RNA-seq data, and this is so it can be used to test structural variant calling tools. So a bit of why this is important is because gene expression is also, can affect your structural variant callers, especially in transcriptome data, as well as just as much as mutations can. So, for example, you get different expression profiles with different tissues, and we wanted to be able to input those as well as the mocked variants so that we can accurately test our tools. There's many different types of tools, and all of these to evaluate them require test data. One of the biggest problems is that you can't even evaluate specificity on real data because you actually don't know the ground truth. So that's why simulating is so important. So how does our tool work? It's a command line tool, and we start with 3 different inputs. We have our annotations, which is like our genes and exons. We have our reference genome, which is the sequences. Well, everybody knows what a reference genome is, but the fastest sequences. And then we also have our structural variant file. So for right now, our proof of concept that we're working on, we're only doing 1, but eventually we'll be doing many different structural variants all in the same simulation. So these get input to our tool where we create inputs for Flux Simulator. Now Flux is a tool that simulates transcriptome data. Normally it just takes a regular genome and our GTF, which is their annotation format file. So what we're doing is we're mutating the existing reference genome and translating the positions of all the existing annotations so that we can then input it into Flux Simulator, and our output is then our faked RNA-seq data. So this is just a little bit like of an example for one case. There's many different types of structural variants. One of the most complicated, arguably, is inverted translocation. So right here you can see that the steps we had to do to create our pipeline was actually first creating these fusion chromosomes and then moving all of the genes accordingly to where they exist now, as well as the fusion that would exist over the breakpoint. This, again, we create these reference files, which we put into Flux, which it then uses the reference annotations to create transcripts, and then it creates the RNA-seq reads from there. So we did not, unfortunately, finish the whole thing, but we are very close. We think one more session and we will have something that is up and working. We've got all the individual components. We just need to put it all together. This is a preliminary result from one of the expression profiles that we're working on. So we wanted to have a bunch of different inputs where you could input different known tissue type expression profiles into your so that you could simulate different tissues that have this structural variants. This is showing that our RNA-seq data that we simulated has currently a 0.54 correlation. Not great, but it's showing that it's better than nothing, and that's our first result. So we are actually generating something that works. And I had also many team members who helped out, which we listed on the front slide, so I'm not going to go again, but I would like to say thank you to them.
I guess our next team is, I just named you infectious blockchain.
Howdy, everybody. My name is Vina Gorkapi, and I am the team lead for Infectious Blockchain. Before I start, I would like to give a big shout out to James Zong and Frank Prick. They are the 2 high schoolers on our team, and they did a lot of good work over this weekend. So thank you so much for participating and having a good, I hope you guys had a great time on our project. And these are the other team members, Anita, Kristen, Kevin, and Zach. We all had a great time working on this project, and we hope you guys will see the work of the application that we developed. So blockchain is it's a list of records that is linked by cryptography, and each of these records is called a block. anytime a block is created, accessed, deleted, anything that happens with the blockchain, all the blocks within this record, within a chain are altered altogether. Each function is, each time the block is accessed, the unique hash function is generated with each transaction. And all these transactions are kept on track of with a group ledger. So a group of people can each have their own ledger, and each of them will be able to keep track of what happens within this ledger. And infectious diseases are diseases that are spread by microbes, so bacteria, viruses, any of these small microorganisms that are used, infectious diseases are spread through them. And right now there's a focus on by the CDC on trying to prevent future diseases from being future outbreaks and future cataclysmic events. But what we want to do is focus on tracking the disease as it's being spread. Data security is not a major focus in bioinformatics, and in the future there will be more of a focus on having secure data that is easy to access and analyze. So the novel application this is going to be a novel application of blockchain. This has never been done before. The closest application of blockchain that is similar to this is MedRec, which is a MIT application that keeps track of different patient records. So it seems like the next logical step would be to use blockchain to track the real-time impacts of infectious diseases. So the goal of this application is to develop a secure application that can be used by members of the general public and by doctors to track how disease spreads in real time. What are hot areas? What are areas that people should stay away from? Maybe location data that might be pertinent. For example, with Zika, people were told to stay away from large bodies of water. So that was done by looking at the data and modeling it with location aspects. So understanding how the data works will be good for both the general public and for people who are in the biomedical field. So there's a promise of quick data access. Right now, these transactions can occur on a scale of seconds. The most I saw was probably like 2 or 3 minutes for a blockchain record. If it was a massive one, like thousands of Bitcoins, then yeah, it took probably a couple more minutes. And secure data access, each time the block is accessed, all the data is changed. And this impacts every node. And correct data access. So everyone's going to have the same copy of the nodes that are altered. So our target groups are medical. Oh, really? Okay. Okay. So I'm sorry, guys. So this is our website, and thank you guys so much for our presentation.
But if you still want to check out the slides, they're all on the presentations channel, so you could also go and like look at those there. I guess now we have our last project, right? Yeah, I guess so. Yeah. Oh no. Do you want to open it like another time? No. Do you use this one? Okay. So, please start.
Okay, hi everyone. So my name is Nushin. I'm the team lead for Project of Mobility with a wonderful team of mostly ladies. We have one. So the background of the project is that there was a mandate in 2016 by the United Nations that for the economic development of the world. And so in Canada, what Canada has done is they've released a series of surveys that include life expectancy, causes of death, and diseases from all of Canada and from the provinces based on gender, disease, and life expectancy. So what we did, and so right now the data is all in large spreadsheets that are not really human friendly and not clean with lots of numbers that makes you get headaches. So our solution is to make interactive user-friendly dynamic charts so that you can make hypotheses and test them right away on the screen and make better informed decisions. So.
Okay, so one of the things we decided to do was make a Shiny app. So we did some data cleaning, and then one of the visualizations we used was this, they call it a tree map. So it basically shows you the bigger the rectangle, the more people that died from that particular cause of death. And then we added some interactivity as well, where you can change whether you're looking at males or females. And then you can also choose the age. So you can see that like a lot of the main causes of death differ a lot by what age you look at. And then you can also look at the data from different years over time. And then we also did a comparison between males and females. Our favorite little comparison on the team was looking at the 30 to 34 year group, where men tend to still die from accidents, whereas the females seem to have gone past that. So yeah, that was one of our favorite things. And then we also put together a map showing by province as well. So Mariam, what can you see on that?
So this map was done with Leaflet, and this map shows you the life expectancy in each province according to, this is the birth year, and then this is the life expectancy, and yeah. Oh yeah, I forgot, I made another map. there's this other map.
So we had,
Yeah, so in this map, you have the option of choosing your sex, and yeah, it just shows the different life expectancies. As you can see, there's a big difference between the male and female life expectancy. I wonder why that is.
Yeah, so these are just some GG Animate plots that were generated using the data. Because things that move and have pretty colors are more engaging than things that are static and are in a table.
Oh, so this is an example of a plotly graph, which allows user interaction. On the x-axis, we have the year from 1980 to 2016, and on the y-axis, we have the age with the highest number of deaths. So we have top blue, which represents the female, and then the bottom green is male, and we can see that males are catching up. And so the slider at the bottom changes between provinces, so users can change. And at the end, we have a word cloud.
That's it.
Yeah.
Thank you so much. I guess, did I miss any presentation? Everybody presented. Yeah, okay, good. Well, thank you so much. So I guess we need just to talk about a few things.
Do we want to do just a survey right now?
Yeah, so we have put together a survey that we would like you to fill now, because it includes voting on your favorite project that you did not participate in, of course. So it's like gonna take you a few minutes if you could fill it up, and then so we could have to know who's the winner here. And Artem, are you sending the survey?
Yeah, it will be on the Slack channel in 2 minutes.
Yeah.
Wait, where is the survey?
you got it. Yeah.
Okay, so now if you'd like to like fill it here, you could stay here. If you want to, if you don't have good internet connection, you could just head out. We'll be finalizing HACKSEQ and announcing the result of the survey. I guess in 30 minutes. Yeah, at 5 o'clock sharp. So we'll be here again at 5 o'clock sharp. So after we come in and we close in, we're just going to go out back to the atrium to have one big photo for everybody in the hall. Okay? So I'll see you in 30.